So we have a few minutes for questions and just pull in a few lessons from pulmonary medicine, respiratory medicine at large for thinking about post-TB lung disease. And I think we've heard a lot about complexity and heterogeneity, and I love Thurnessen's message of embracing it. And I think actually we have to not just embrace it, but anticipate it. It is going to be there, and so we should plan for it so that we're not surprised on the back end. I think there are some success stories and cautionary tales from pulmonary medicine that we can learn from with phenotypes and endotypes as they've arisen in asthma and ARDS, or the acute respiratory distress syndrome. And then also some lessons that over the last several decades we've learned about treating idiopathic pulmonary fibrosis, where we have to be sure that we really are indeed addressing pathogenesis. And so just to be sure we're all talking about the same things, a syndrome apparently comes from the Latin of syn with and dromos, running, so it's a running together. It's a, we wave our hands and say, oh, these people are all kind of the same. Versus a phenotype, which is something that's sort of a flavor of a syndrome. If you have a broad disease, what are the different flavors that you have? And then the endotype in this context is the biology that underlies those flavors. So what's contributing to the different flavors that we have? What are the pathways and mechanisms under that? And certainly in the world of respiratory medicine, this really, I think asthma led the field. It used to be that we would just call everyone with asthma the same. And this study, which was done in Colorado, published back in 1999, they found that you could really divide asthma up on the basis of eosinophils. And so these allergic cells, people had really different clinical characteristics. Their propensity towards severe exacerbations was different. And that this correlated with subbasement membrane thickness in the airways and otherwise. And this led to a sea change, a real paradigm shift in asthma care, where now we routinely try to classify patients as having T2 type eosinophilic flavor of asthma versus non-T2 type. This has been really well delineated, and this beautiful figure was published in the New England Journal a few years ago. And gosh, if we could only have this for post-TB lung disease, this is what we want to aim for. But I think it really nicely highlights that these distinct molecular pathways that contribute to these different clinical flavors, these different phenotypes. In the asthma world, they call what we call host-directed therapies, they call biologics, and there's many of them that have come out. The first was approved back in 2003, and they've continued to have sort of more and more over the years. I'll mention just for a moment one that's called dupilumab that acts on IL-4 receptors. And the study that led to dupilumab approval, you see the main results from that here. They enrolled patients with severe asthma, and we're looking for severe exacerbations as the primary outcome. And you can see overall, there was a benefit in the entire study population, a 48% reduction in the risk of severe asthma exacerbations. But when they broke it down by eosinophil count, by which phenotype or which endotype of asthma you had, the benefit was really limited to those with the high eosinophils. So just emphasizing and underscoring that if we understand the biology, we can better direct our treatments. And then the next quick story is about acute respiratory distress syndrome, or ARDS. About 10 years ago now, there's a group out of San Francisco that led latent class analysis and identified these subphenotypes, these flavors of ARDS, instead of us just saying everyone is the same. And they found that based on a data-driven approach towards looking at both biomarkers and clinical features, they could define these two subphenotypes, hyper and hypoinflammatory, just as we heard from Leopold, that there likely exists in TB as well. Those who are hyperinflammatory, higher biomarkers, higher use of vasopressors, higher mortality. And so why does this matter? They then went back to what had been a previous study of simvastatin in ARDS. It had been a negative study. They recreated their subphenotypes and found that actually in the hyperinflammatory group, it appeared that simvastatin was effective. And so when they combine all the patients together, you lose that signal of benefit for some of your flavors, some of your subphenotypes. And those are the two red lines there versus the hypoinflammatory, where there was no difference by simvastatin. Similarly, in COVID-19, they also found these subphenotypes existed. Again, mortality was worse for the hyperinflammatory. And here, and this was retrospective, they found that corticosteroid responses were different. So we were giving everyone with COVID steroids for everyone. But you see here, for those who were hypo-inflammatory, mortality was actually higher if they had gotten steroids versus the hyperinflammatory was lower. And so I think in the context of TB, we want to be sure that we're anticipating these flavors so that we're not losing signals of benefit that might be helping some patients but not all. And then very quickly, because I want to try to have a few minutes for questions, in terms of idiopathic fibrosis, pulmonary fibrosis, which is a chronic pulmonary disease, has features that Thrinessen had mentioned, like fibroblastic foci, and historically, we haven't really known what causes it, which is why we call it idiopathic. And so back around 2009, most patients were being treated with immunosuppressants. We said, oh, there's something bad is happening, we're just going to throw steroids at the problem. And so the large trial that looked at this was the Panther IPF trial. It was targeting inflammation, and so patients were given a combination of prednisone, azathioprine, and NAC, and they actually found that there was an increased risk of death in those who were receiving what was considered the standard of care at the time. And so people said, oh, maybe NAC alone would be okay. They actually didn't find a benefit from NAC for lung function, but again, digging into it a bit further and sort of getting at the idea of some of these things, are they benign therapies, or do we need to be looking for some of the subflavors? They found cardiac events were a bit higher. And then actually, there's a host gene polymorphism in IPF that impacted whether you had benefit from NAC or actually had worse outcomes. So again, I think across all of these diseases, we see that there are flavors and these flavors matter. And so historically with IPF, we thought it was an inflammatory process. We used anti-inflammatories. That killed people. It made them worse. Now we realize it's more about ineffective repair and our use fibrotics. And so just closing for TB, I think as we've heard so many times, we know lung disease is heterogeneous. There are all these different flavors. There's this dynamic balance of damage and repair. And so being very specific, what patient are we treating? What process are we trying to address? What time point are we trying to deal with this? And knowing what outcomes we're measuring. And hopefully, as we've seen in asthma and IPF, with the persistence, we'll start to identify what are the really positive interventions we can make for our patients.